with that, I would like to introduce our last speaker for this morning, which is Gerd Maass. Gerd Maass is the head of the Tumor Profiler project from Roche. And yeah, he will present us the project, which is about multi-omics tumor profiling for clinical decision support.
Yeah, wonderful. Good morning, everybody. Can you hear me well before I start? Okay. Yeah, the test was good. It's still good. Wonderful. So listening to the presentations from the to what Olivier explained, we are diving now a little bit deeper, and it's going to be a lot of detail. And I try to keep it high level, but if you have questions later, because we really dive then into the technologies. I'm going to open my presentation now, and just hoping that that works as well. Good. All right. Just 2-3 slides for the introduction. I'm working since 20 years in this field of personalized healthcare, and one of my first experience was really working on Herceptin, the breast cancer drug. And we had early on findings with this drug that some of the patients really responded very well, long over time, while others relapsed early. And it was very complicated to have animal models or whatever to try basically to simulate that. So this was a typical example early on where we tried to have access to data from patients. We designed several trials. We went into the earlier onset of the disease, earlier treatment, long trials, thousands and thousands of patients in clinical trials. So this was one of the first examples where basically access to good real-world data, basically patients' data, could have helped a lot early on and maybe brought this medicine to many more patients. I just wanted to emphasize that because that was 15 years ago and it was very obvious. The second one is that we are all aware basically that the individual data rights, should be very considered very, very safe. We have to be safe here what we are doing. And when we look then into the different types, it's perfectly fine sometimes if you, for example, validate a diagnostic platform, if you don't have a lot of patient information, because in this case, it's probably really the tumor, the tumor type, the tumor load, which can help. While if, for example, if you look for medicine and how medicine really act over time, of course, you need to have access to certain data so that you can follow longitudinal. And this is basically where really the discussion starts very often, how you can do it. But this is where the real value, on the long term for the development and extension of medicines will really lie. One more slide to the more general concept. We do that. As Roche, we have a deep data protection expertise. We do it over many, many decades, as I said, with large studies all over the world, thousands of clinical trials, millions of patients involved here. And then, of course, you also have then the marketing studies. But the one point on the very right side, I want to explain today a little bit more our collaborations with data partners on R&D and patient care and access. This is basically the start why we, a few years ago, had very deep discussions with some partners in Switzerland. They are listed here. So University Spital Zurich, Zurich University, ETH, and then the University Spital from Basel. And we said, okay, we want to do a partnership which will aim to improve cancer patients' lives through personalized cancer care. So it's really about patients. Right from the beginning, we said the aim is really to try as soon as possible to work with real data, patients' data, and also to improve actionable clinical decision-making so that we have data in our hand, which can really lead to new treatments and to new recommendations. And we said, okay, and this is now really the big part. In the beginning, we tried to collect different technologies. And all these technologies together should give a better understanding on the patient, on the tumor, on potential treatments. So, and each partner brought a certain strengths to the program, starting here on the right upper side with the ETH, where it's world-class research institution for molecular system engineering, broad expertise in omics technologies, data science, machine learning, computational biology, and bioinformatics. then the University Spital Zurich with large clinical trials. We had this initial focus here really on melanoma, but then also on blood cancer. And we already had a partnership. We jointly developed a sequencing laboratory. It's the Foundation Medicine Technology. And that's the only one in Switzerland today. Then University Zurich, very good research, biomedical translational, biomedical digital sciences, cutting edge omics technologies, very complementary to ETH. University Basel. Here we focused at the beginning more on the gynecology. We'll talk about that, ovarian cancer. And then of course Roche with the diagnostics, the pharma, and all our R&D capabilities. And if you look what we really designed then, is if you look at it on the left side, that's the current routine in Zurich. So FMI, that's already a fully validated next generation sequencing. Then you have, of course, immunohistochemistry, but also digital pathology, which currently results in the standard clinical report, which goes to the tumor board. And now we said, okay, let's try to complement it with the right side, where we do a very comprehensive tumor analysis on the DNA, on the RNA, on the protein, on the cellular, on the tissue level, and also check drug response, which then will be a molecular summary report from molecular tumor board, which then will also go to the tumor board together with a standard tumor board, and then you can compare from our current routine what kind of recommendations you can make for a patient, and then you add the additional information from the right side, and then you bring it together and see how much these new technologies can help here in the beginning to bring new treatments on the table. And we started that in 3 trials, and they are all ethic approved trials, but obviously these are not interventional trials, as you would need all of these technologies fully IVD approved and developed, and that's at this stage not the case yet. But we got these observational trials, but we also got some permission from the ethics if you have really interesting and meaningful results that you can do healing attempts, so Heilversuche. You can treat patients which are running out of options. And so that was the concept. We started that, and we said, okay, the specific goals now going a little bit deeper. So more sophisticated, comprehensive molecular tumor boards. And then, of course, we checked here collaboration across hospitals and departments, which all of you know, it's not easy. If it's about technologies, about data sharing, clinical decision, joint tumor boards, and on the R&D, research and development side, to improve correlation between response markers. So and that's really beyond the standard genomics of next generation sequencing. And very important, tumor heterogeneity, handling also by including single cell sequencing, and improve technologies as it relates to the indication. So we have the first examples here, and you really get very interesting results if you look at this. And maybe to go a little bit deeper even, so the way how we set it up was that we have a central lab where the tumor, basically it's all fresh tumor, I have to mention that. These technologies at that point cannot work with paraffin embedded, formalin fixed tumor tissue, which is normally the case in the routine standard. At this stage, we still need access to fresh tumor material from every patient. And this team really, succeeded in getting an infrastructure in place where every tumor goes to a certain central lab. And from the central lab, they ship portions of the tumor to different parts. Just to give you an idea, there are more than 100 scientists involved in this matrix. And we had at the beginning the goal where we said, because we really want to mimic and show the real situation, 4 weeks is the maximum for the whole diagnostics analysis of the tissue and the reporting. So if you go and think about a bi-weekly tumor board, so at least after 2 tumor boards from a patient, you have the full result. And I have to say, really almost to my surprise, this matrix team from the different departments managed in almost all patients to get the full data report filed and to the actual tumor board within 4 weeks from the time you take out the tumor of the patient. Very interesting. Just to give you an idea what it takes here. So we had the opportunity identification in 2016. And of course, you can imagine a series of meetings was necessary to get to an alignment and preparation for this partnership. This resulted in a contract for all of us. Ramp up and validation, that was in 2018. And then in 2019, we started the clinical trials, as we said, the ovarian cancer, the melanoma, and now also blood. And we expect the results towards the end of 2020, early 21. Unfortunately, we had about a 3-month shutdown. That was the Corona impact earlier this year. So we probably prolong the program a little bit more to collect the full data set of the 240. All of this is currently in discussion. And from the data we are seeing, I'm not prepared basically to show results. It's too early. But what I can mention is we see really interesting data here coming out of the different technologies. And it's opened up doors really to new treatment options. So that's for the future really interesting. Now the whole analysis, you have to think it the way that normally you do the genetics. But as we all know, genetics is based on DNA, and it's a kind of indirect correlation to treatments. Because of very late data sets where you see it, you're never sure in a single patient or in a tumor that what you see on the DNA level is fully translated into the biology. And those technologies really, you know, give a wide spectrum from a large tumor to small tumor pieces to single cells, to RNA activation, to protein activation, to drug-drug interaction. So that this full path from genotype to phenotype is covered, and we are getting the best of all the information now basically to make a meaningful recommendation for the tumor board. And you can imagine that this machinery here, from the clinical, the pathology, genomics, drugs, proteins, you know, to an integrated data, to get to the phenotype and to the computers. This was a huge exercise and still is, but as I said, it's working quite well. And if you think about the data flow, it is really a digital ecosystem across institutions. You have the R&D technology nodes, they have to report into a system. You know, the system goes to the pre-tumor board. And at the pre-tumor board, you have to condense this to really meaningful information for the actual tumor board, because the clinicians know that the tumor board per patient might spend 10, 15 minutes, and very often it's already overloaded with the data you get out of the routine. So the art here is really, you know, the challenge to bring this, all of this information from these R&D activities through the pre-tumor board to a one-pager, which really shows what of this technology can help for a decision in the actual tumor board. And this is my last slide, but that shows, I think, very nicely the challenge where we are in the middle and what we try to achieve. So the raw data, it's huge. And then you go down to the condense it a little bit more to all results from the R&D nodes, then for selected clinical reporting. And from the selected clinical reporting, you try to be predictive. So it's more than the medical people, and then to the relevant data sets from the pre-tumor board, which then will be discussed together with the tumor board. So from 500 gigabyte to 1 megabyte information. And this is just what we are in the middle of it. We will probably need another half year after this trial is over until all of this data will be analyzed. And then we need to see basically how this can be made broader available, or where also the limits are, because it's just too many data. And which data of those data sets that are the most meaningful for the future, for future trials. So it's a bit too early to talk about it, but I want to say a very exciting program, and the interaction with the scientists, with the medical people, this is unique. I've never seen it before. I was really positively surprised that 1 year after we discussed this program, you know, we see this flow, so that tumors were distributed, data were collected, and tumor boards could really use this kind of data in their discussion. Thanks a lot. I think that was my presentation, and happy to take questions.